Kia ora koutou. Ko Sue Brewster, taka wingua. Well, good evening, everybody. My name is Sue Brewster, and I have the immense privilege of being the Executive Director of Auckland Medical Research Foundation. For those of you joining us for the first time tonight, welcome. And our foundation was founded in 1955. So next year will be our 70th year anniversary celebrations, which is quite some achievement. We are the largest independent funder of medical research in New Zealand, and we fund across the whole spectrum of diseases and conditions. And one thing that's really unique about us, and I know that a lot of our donors love, is that 100% of every donation of every single cent that we receive in income goes straight into our mission, which is funding medical research right here, right now. So tonight we have the immense privilege of having Professor Peter Thorne and Dr. Haruna Suzuki-Kure presenting. So Professor Peter Thorne is an extremely busy man. So he is with the Department of Physiology and Department of Audiology at Auckland University. He was also a member of our Volunteer Medical Committee. He is a trustee on our board, and he is a director of many boards and other trusts. His specialty is in audio science, the audio neuroscience, and he has spent many years looking at the ear, the inner ear, diseases affecting the ear, how treatments can help preventing hearing loss. So really knows so much in this field, and we're extremely fortunate to have him here tonight. Dr. Haruna Suzuki-Kure is a research fellow at the Department of Physiology at the Auckland University also, and her latest research is looking into those tiny little blood vessels that feed that inner ear and how changes to them might affect our hearing or contribute to hearing loss. And this is all with the aim to develop new therapeutics and treatments for hearing loss. So we're very lucky to have them here tonight. And Professor Peter Thorne will open the evening, then followed by Dr. Haruna Suzuki-Kure, and then Peter will do a wrap-up summary. So that should all take about 45 to 50 minutes, and then we will have time for questions and answers, live question and answers. So stay with us, and you can be feeding in your questions into that live chat as we go throughout the presentations tonight. So please sit back and relax. Don't forget, keep putting your questions in, because there will be that time at the end to ask those questions of Peter and Haruna. So without further ado, I'll hand you over to Professor Peter Thorne to talk to us about hearing loss and diseases of the ear.
Tēnā koutou katoa, nā mai haere mai, ko Peter Thorne toku e noa. Hello, my name is Peter Thorne, and welcome to this AMRF webinar where Dr. Haruna Suzuki-Kure and I will talk about some of the research on the inner ear that we're undertaking at the moment, and looking also at some of the development of future therapies, which is why we called this what might the future look like. So we're going to do this webinar in three parts. In the first one, I'm going to talk about ear diseases and hearing loss, and think about how this occurs, and think also about some of the treatments and interventions that are available now and are on the horizon. Dr. Suzuki-Kure is then going to come and talk to you about some of her AMRF-sponsored research on the vasculature of the inner ear, a very important area, to finding new treatments for hearing loss. And then I'm going to come back in again and just give a bit of a wrap-up, but talk a little bit about some of the work, or additional work that we're doing in terms of some of the programs that we have. So let's just talk first of all about hearing loss. The first thing here, which is presented here through a report which was published by the World Health Organization in 2021. It was the first world report on hearing, which is a global report on our understanding of hearing and hearing loss, and public health as a public health issue in particular around the world. And so this gave us a lot of data, the most important, which is around about 1.5 billion people around the world are affected by some form of hearing loss, and that the cost of that to society is close to $1 trillion annually. And so this is a big health issue. And as you'll see in the graph on the right-hand side, the situation in New Zealand is similar in terms of proportion. And we believe around about 800,000 people in New Zealand have some form of hearing loss. And it costs the New Zealand economy somewhere in the order of $4 billion annually. So let's have a look and see what this actually means. So hearing loss comes in all sorts of different grades, and from mild through to a profound loss or complete loss of hearing. And the majority of people with some form of hearing loss have really mild to moderate hearing problems. And what does this actually mean in terms of function? Well, first of all, those that are in the sort of mild to moderate area will have difficulty in hearing conversations in particular in noisy environments. But as you get into the moderate to severe or profound losses, then it becomes far more of an impact. And so people will have difficulty in hearing when there is in groups or within background noise. And as they get worse hearing, and so they will be not able to understand speech at normal levels, and often when it gets to profound, not even be able to hear that speech at all in very loud sounds. So the impact can be very, very high when you get into those areas of severity. So let's just think a little bit about our system of hearing, our hearing system and our sense of hearing. I've got a diagram here which just shows the ears and the brain, and just to show that the fact that these are obviously all connected. The ear is the portion that most people will know about. It contains the areas of the ear, which I'll talk about a bit more later on, which detect the sound and then produce impulses in the nerves that go to the brain, and particularly through the brainstem, which is shown here as number 7, and then going up to the cortex, the auditory cortex, that part of the brain which is responsible for making sense of the sound that comes and the information that comes from the ear. And the cortex, the auditory part, is actually out here on the temporal lobe, this part of the brain. Now, this slide is actually very important because it shows the two ears. And whilst in most diagrams we just look at one ear, it's really important to realize that the ears work together. And they work together in order to be able to localize sound in space, but also to be able to help us to understand speech in background noise. And so it's important to realize that people who have maybe just one poorly functioning ear, actually also have quite a serious disability as well, and will have problems, particularly in background noise. So even a single what we call a unilateral hearing loss, is important to treat. So the ear is in 3 parts. The first part, which is the portion that you often see, which is the pinna and the ear canal. And then there's the middle ear. Those 2 parts are separated by the eardrum at the end of the ear canal. The sound passes down, and it then will cause the transmission of the energy across the little bones inside the middle ear. The middle ear is just a cavity inside the skull, which is filled with air, and that connects with the back of the throat through the eustachian tube. So it maintains the air pressure across the eardrum. The sound is then transferred, as is shown in this cartoon here. The sound is transferred across the middle ear by the little acicular chain, what we call them, all these small bones, which are the smallest bones in the body, and they transfer the sound into the inner ear where all the detection systems are, and enables us to detect that sound and send impulses to the brain. So often hearing loss is categorized according to where it occurs inside the ear. So we often think about hearing losses that might occur from changes in the outer ear or middle ear as what we call conductive hearing losses, because these losses occur because the apparatus, the ear canal might be blocked, for example, by wax, something as very simple as wax, blocking the ear canal, or diseases inside the middle ear, such as glue ear or infections, which lead to the change in the conduction of sound into the inner ear. So it's a conductive hearing loss. Much, much easier to treat, by the way, than the sensory neural hearing losses, which I'm going to talk about now. So hearing losses that occur as a consequence of injury to the inner ear, the part where the sensory cells and the nerve fibers exist, is often called a sensory neural hearing loss. And here, this tends to be permanent losses, as opposed to a lot of the middle ear ones, which can be well treated or might be temporary. This is caused through maybe genetics, lots to do with noise exposure in the environment or at work, drugs that might cause the degeneration of the sensory cells, trauma, such as head injuries, infections, a lot of inner ear diseases, which are like Meniere's disease, for example, some of you might have heard about. And one in particular, which Dr. Suzuki Kerr will talk about, is this thing called sudden hearing loss, which is where a person might develop a hearing loss suddenly or over a very, very short period of time. And of course, a lot of hearing loss comes as a consequence of aging, so we call it age-related hearing loss. And of course, some of the changes, these changes in the ear, will cause hearing problems or associated problems in the brain and degeneration or loss of nerves inside the brain. But there are also processing problems or disorders of hearing that come because of changes inside the auditory parts of the brain. And we often call these auditory processing disorders, where a person might have very, very poor speech understanding, or speech might be completely jumbled. All the sounds will be all mixed together, so it's very, very hard to interpret that. And of course, tinnitus, which is the presence or perception of sound when there is no sound in the environment. And we know very little about how all of these brain changes occur to cause hearing loss. The ear disease, hearing loss and ear disease might occur across the entire life course. And we can see that there are factors that might occur during the prenatal period, perinatal period, for example, meningitis. There might also be events that occur during childhood, otitis media, for example, which is the hearing loss that occurs from glue ear. And then I've talked about medicines such as ototoxic medicines and so forth. Then some of them can be prevented, actually, to some extent by, for example, vaccinations, maybe good nutrition, looking after your ears and preventing it from being exposed to noise can help prevent the hearing loss. Now, the majority of hearing loss, and particularly as we age, as shown in this graph here, which is shown by the blue histogram there, is actually due to sensorineural or inner ear diseases. And I'm going to spend some time now talking solely about what we call, as we've already described, sensorineural hearing loss. So let's have a little bit more of a detailed look about the inner ear. And so this diagram actually walks you through from the curly part inside the bone, called the temporal bone, called the inner ear, which contains both the vestibular or balance organs, as well as a structure called the cochlea, which is the hearing organ. It's just really a series of tubes that spiral within the bone, and those tubes are filled with fluid. And just like a snail shell, if you like, they spiral around a central column, and that central column contains all the nerve fibers and the blood vessels and everything that comes in to support the ear. And it goes out to the sensory organ, which is situated inside the fluids and spirals around as a ribbon of tissue, around about 3.5 centimeters long, spiraling around the central column, which we refer to as the modiolus. And if you look at that tissue or the sensory organ in more detail, you'll see that it's made up of a whole lot of sensory cells, which we call the outer and inner hair cells. These are specialized cells, which, as you see on the left-hand side, have little sensory hairs that project from their surface, and it's those sensory hairs that bend backwards and forwards with the vibrations and lead to the stimulation of nerve fibers, which sit at the base of the cells. And this is just what a human inner ear looks like. This is taken from a slide, from a histological slide of the temporal bone, and you can see what you might take, if you imagine taking a snail shell and cutting it right down the middle, or even a periwinkle or something that you get off the beach, cut it straight down the middle, and you'll see the cavities and that what appears to be a spiral structure going around that central column that contains all of the nerve fibers. And they're called, in that cavity area, they're called the internal auditory canal. Now consider that as I was saying, that the sensory organ is in a ribbon that goes around that central column. And that's shown here on the left-hand side using a special microscope called a scanning electron microscope, where we've removed all the bone around and taken the inner part, the sensory part, out of the bone, and you can see that ribbon of tissue with what look like a whole lot of little seagulls or something sitting on the top of it. They're little white specks. They're all those sensory hairs from the sensory cells. And what's really important is that all the cells are slightly different along the ordinal cordae, and they respond to different frequencies or different pitches of sound. And so the ones in the base, right down at the base of the cochlea, they respond to very high frequencies. The speech sounds that you might have as consonants like a T or a S. And as you go further and further up to the top of it, what we call the apex, then they respond to lower frequency sounds, right, which are the vowel sounds. So we call this a tonotopic map. It is a map, a spatial map of the tonal representation of sound. So if you have a hearing loss in the high frequencies, it's going to be due to damage or changes that occur in the basal part of the cochlea, compared to one where you might have a low frequency or low pitch hearing loss, where it's going to be affecting the ones up in the apex. And this injury is due to sorts of changes that you see here: loss of the sensory cells, loss of the little sensory hairs, and in the bottom one there you can see on the left all those areas, the nerve fibers, and as shown by the red circle, all those nerve fibers are missing because of diseases, and that's taken from a human inner ear, and that person would not have been able to hear the high-pitched sounds represented in the basal part. Now, we know a lot more about the mechanisms of the injury, and we're still learning so much about it, and we need to know and understand this in order to be able to develop targeted therapies to treat hearing loss or restore hearing. And a lot of it's about how can we target the inner ear and either replace the cells or prevent them from dying, prevent their injury, and provide those sorts of treatments. And we now know there are a lot to do with genetic disorders, vascular disorders, which Dr. Suzuki Kerr will talk about, even inflammation, inflammatory responses, which you might be familiar with. Your knee gets injured or you cut yourself. These inflammatory responses can also occur inside the inner ear and lead to damage and loss of the sensory cells. So what are the therapies available? Well, you'll be familiar with hearing aids, which are very important to provide amplification to those areas where there may be just a few cells working, or if there are no cells working, providing the sound amplification to other areas of the inner ear. Cochlear implants, which is a device that's placed into the inner ear where the sensory cells are missing, and it will cause the nerve fibers that are still present to fire. And the important thing about the implant is that it will activate the nerve fibers in the base of the cochlea for high frequency pitch or high pitched sounds, compared with the ones in the apex where it's going to be for the low pitched sounds. But what about molecular or gene therapies, which is the area which we're working in substantially? And at the moment, there is no effective drug that is approved for use for sensory neurohearing loss. So this is a huge gap. And this is why we're particularly interested in our research in finding ways to develop drug-based therapies or gene-based therapies to prevent or reverse the pathology in sensory neurohearing loss. So looking into the future, what are these cochlear therapies looking like? Well, let's just think about some of the things that sit behind the development of therapeutics at the moment. First of all, from a lot of animal work, a lot of the research which we and others all around the world are engaged in and have been for many, many decades now, leading to a greater understanding of the causes of cochlear damage. And because we can measure the hearing loss in animals as well, we can see how that damage actually relates to the hearing problems that might be similar to what people have. The research, though, is limited to animal studies, and we don't have a lot of information about what goes on in the human in the ear, which is important for us to develop those therapies which are going to be effective for humans. We also have, from a lot of studies, and we've done quite a few studies now looking at particular drugs and compounds which we believe have a therapeutic effect. And we have a lot of that preclinical evidence. That is evidence that is in animals that shows that if the hearing that you can restore or prevent, and in some cases with gene therapies, to actually recover or regenerate the sensory cells or the nerve fibers inside the inner ear. So there are effective therapies within animal models. And because of this, there are a lot of companies now formed to actually develop therapeutics in the hearing space. And there's quite a lot of trials underway across different countries, and either single or multi-center trials to try and see if these compounds have any effect on humans. And so far, it's not been particularly successful. The different sorts of therapies that we can talk about, there are ones which are small compounds, or what we call biologicals. These are ones which are drugs, if you like. They're chemicals which target a particular type of disease. They can possibly prevent hearing loss from occurring, like we might have ones which are, for example, preventing oxidative stress. So you might be familiar with antioxidants, the possibility of using those as compounds to prevent hearing loss. But they may also help in the generation of new cells, or also try to prevent the cells from dying. Then there are gene therapies. Can we, now knowing more about the abnormal genes, can we actually possibly change that or reintroduce some of the products, the gene products, and stop the loss of function, or save the sensory cells or nerve fibers from dying? And there's a lot of interest in possibly finding those genes which are important for how new hair cells are made from the existing cells inside the inner ear. And can we then do something with that knowledge to perhaps promote the regeneration of hair cells, new hair cells, from the existing cells within the cochlea, in a damaged cochlea? And the last one is really what we call cell-based therapies. These are a bit like stem cells. If we understand more about the stem cells inside the cochlea, you might be familiar with stem cells, which are like originating cells. They can be transformed into all sorts of different cells in the body, and they're present throughout the body. And if we can stimulate those or introduce them into the inner ear, then perhaps we could replace some of the damaged cells inside the inner ear. And there's a lot of very, very exciting work going on into all three of these types of therapies, which we hope will be translated eventually in some form into human therapies. But there are big barriers and gaps, and a lot of research is needed to be able to do this translational work, to take what we know from animals to get into humans. So in a way, there is a revolution happening in the lab, which is going to lead eventually, and that's why we talk about what's in the future, what's on the horizon, is that there are things happening in the lab, but there are barriers to that translation. So we do need to understand more about the human cochlear disease processes. Are they the same as what occur in animals? Do we know what happens to cause the death of the sensory cells? Because we need to know that in order to be able to target the actual specific disease process to try and prevent the hearing loss from developing. The next thing, which is quite a significant barrier, is how do you get those drugs or those therapies, whatever they might be, into the cochlea? And as we've talked before, and Dr. Suzuki Kerr will talk a lot about the fact that the cochlea is embedded in the temporal bone, and so it's difficult to be able to get to those tissues inside the temporal bone because of its location. And lastly, how do we know what the disease is going on inside the inner ear? You might have a hearing loss. That's something that we can test, say, using an audiogram. But what do we know? The hearing losses that occur from different different diseases might be the same, but we don't know what's actually occurring in that person. Where is it? Where is the disease actually occurring? What type of disease? So we need to have some way of diagnosing that, some way of peering into the inner ear and identifying what the disease process is. We call this sort of, if you like, we need biomarkers of the individual pathologies in an individual person. So I'm going to leave it there and let Dr. Haruna Suzuki Kerr talk to you about some of her really exciting and novel research into the vasculature of the inner ear.
Kia ora koutou. Nō mai haere mai. Ko wai au, ko harina taku ingoa. Kei wai papa tōmato au ahau, ka e noho mahi, hei kai rangahau. Welcome everyone, and thank you so much for joining us tonight. My name is Haruna, and I am a research fellow at the University of Auckland. My research journey started here in Auckland as an undergraduate and postgraduate student at the University of Auckland, and through different positions, I have joined this hearing and auditory research field in late 2018, and at different time point was supported by Auckland Medical Research Foundation, and I am very grateful for that. As Peter mentioned in his talk, there are still a lot of work to be done in this hearing and auditory field to make improvements and provide different options for hearing care. And since joining this field, I met a lot of amazing people, like really amazing researchers, both clinical and non-clinical. Some doctors, for example, researching different ways of managing inner ear diseases, and audiologists and other population health researchers tackling the problem of limited resources or inequity in hearing care. So it's really been quite an inspiring journey, and I am privileged to be part of that collective approach that a lot of group of researchers are doing to improve the overall hearing health and the hearing healthcare system in New Zealand. For today, as a biomedical science, what I do to contribute to the collective approach in improving hearing care is to conduct a lab-based research to provide a proof of concept data that's new and support development of something new or different ways of treating or diagnosing hearing loss in the future. So that's what I hope to do in the long run. And today I will be giving a seminar from my end titled Making the Roadmap, How Does Blood Travel in Human Cochlea, to share some of our latest research. So just to acknowledge, the content of my part of the talk is purely based on a research project funded currently by Auckland Medical Research Foundation. The project started last year and will finish next year. So I will be delivering and sharing with you the hot off the press data today. And here are the acknowledgement of associated investigators and amazing advisors contributing their expertise for this project. So my seminar is about vasculatures in the area of the cochlea. And vasculature is a term for arrangement of blood vessels that exist in different parts of our body. Now, as many of you are aware, our body is full of blood vessels, and they constitute a network that's a little bit like our road system and a highway system. And this very efficient road system driven by the heart, drives different nutrients and other things necessary for our living in both normal conditions, but also in the times of stress. Now, shown here is a diagram of vasculatures of the brain. So even a large organ like a brain is full of blood vessels to supply enough nutrients and energy for the large and complex system. Shown on the right here is the cochlea and how it receives the blood supply from the same arteries as the brain. So this part here on the right, so this is a cochlea in the middle here, and over here is a brain stem. So it's this central part of the brain, and cochlea share its main arterial supply with cerebellar artery. And as this branch comes into the cochlea, it becomes the Rabin-Weiss artery. As many of you may be aware, a lot of brain disorders have causations or at least some of the pathologies attributed to blood vessels and its change. So, for example, stroke. But even in other neurodegenerative conditions, latest research is showing huge contributions or involvement of changes in the blood vessels as a contributing factor for disease. On the contrary, though, we've never almost heard of vasculatures or blood vessel change being associated with hearing loss. And part of that may simply be that we think it just hasn't been studied. There are some form of hearing loss where the involvement of blood vessel abnormality is suspected. So, for example, with sudden onset hearing loss where you lose hearing all of a sudden, some people have described as possibly being a mini stroke in the ear, where the blood supply is depleted and you lose hearing loss because of that. But other evidences are scarce, and it's partly attributed to the fact that we still don't know very much about how cochlear blood vessels and the vasculature system works in the cochlea. So that's a research gap this project is trying to study. So the research gap for us is there are still unknowns about the cochlear vasculature. The cochlear system, I think it looks a little bit like a shell, or in this context here, like a mountain. And just to put it in an example, we know from previous research where people or important part of the cochlea live in that mountain, from neurons to hair cells. But we have no idea how these people live. How do we deliver meals to those people, or how do these people receive supplies, which has tremendous importance for their well-being. Now you might ask, are you sure it hasn't been studied? Well, some parts have been. And this schematic diagram here shows the blood vessel arrangement in this outskirt part or near surface part of the cochlea. And what it shows is a very complex layers of arteries arranged in this area. And this is based on small animal studies, and these areas, because it's on the outside, they are slightly easier to study. However, there's a huge gap in knowledge about how the vessels exactly travel inside this main compartment to reach out for these different areas within the cochlea. So that's what we want to study. So the research goal for this project is very simple. Our goal is to make a roadmap to understand how blood travels to the target destination in the human cochlea. And in the interest of this talk, I'll be dividing this to part 1, where we are aiming to map blood vessels in the whole cochlea using a technique called micro-CT. And part 2, where we'll dig in a little bit more detail in the selected areas using microscopy approach. So why is it that it's so hard to study the cochlea? Now, first thing first is that cochlea is located deep inside our head, and it's really close or underneath the brain. It's also small, so cochlea is a size that would sit on my fingertip like this, and we often also describe it as being about the size of a pea. So it's a very small organ. Naturally, the vessels and structures are small inside. And finally, the cochlea is inside a very hard bone, like a rock. So the cochlea is situated inside this part of the bone called temporal bone, and it's one of the densest bone in the body, just like the rock. So in a way, wanting to say we want to study vessels inside the cochlea is like saying we want to study tiny vessels inside a rock. And you can imagine that being very, very difficult. So this means that our team's mission is really how would we study a structure that's so small and complex and it's inside the rock in 3D. And that's what we are embarking to do. So now many of you may have heard of a technique called CT scan or may have had a clinical CT scan. So when looking inside the bone or inside a large body, CT is a useful technique It's X-ray based imaging, where X-ray penetrate right through your body and form an image, and you take thousands of them to construct a 3D volume of images. And micro CT is a variation to CT that it's micro. It focuses on a very small tissue and provides a lot more detail. So the sample for this research comes from a post-mortem human tissues donated to the Human Anatomy Lab at the University of Auckland for research and education purposes, and we use part of that for research. So sample is human bone, and after X-rays imaging, it generates a series of cross-sectional views throughout the temporal bone, which we can then 3D reconstruct as a 3D volume of images. And the beauty of this approach is you can take a cross-section anywhere you want in an angle to look inside and now analyze inside. Like this example here, where we can clearly see the cochlea inside this hard bone and start to investigate where the vessels might be and how are they traveling inside the cochlea. So this is an image of micro CT taken of a cochlea, and I've taken a cross-sectional view in the middle, looking like a figure here. And what you see is it's a black and white image, and white part is the bone. So cochlea is full of bone on the outside, which has been trimmed, but it's also full of bone inside. And based on previous studies, we have full understanding of where the important hair cells or neurons live inside this structure. But where are the blood vessels that we are looking for? If we zoom in, this is how it looks. In the central part of the cochlea, which is full of bone, you see a lot of different cavities or holes in the bone. Now these are the holes which houses or contain the blood vessels within. So what we have in the cochlea is this situation where even inside is full of bone. So for blood vessels to travel through the bone, we have tunnels. Now we call them bone canals, but we have the tunnels. And what we are visualizing here with conventional micro CT is the tunnels that contains the blood vessels. Now this is great. We can map the tunnel network inside the cochlea, which will give us some information about the blood vessels. But what we really want to know is where the actual blood vessels are. So how can we visualize the blood vessels themselves instead of the canals, bony canals in 3D? And this is why we have to employ and apply a new technique. And what's different this time is we've got a special dye from scarlet imaging, and it's like it looks kind of just like a paint, and we have injected this paint into the blood vessels of the cochlea beforehand, and we go through the same sample preparation procedure and scan the image with X-ray, micro CT, and reconstruct the image in 3D. But this time, if we take a cross-sectional view here, what you end up here looks like this. So we still have the bone now shown in blue color here, but now the blood vessels themselves are filled with a stain or the paint, and that paint can be visualized as this yellow colored signal in 3D volume. So this image is almost looking at about entire cochlea, showing blood vessels in yellow color. Now this approach is very new and it hasn't been applied to the cochlea in the past. So we're very excited that the approach finally worked. So this is hot off the press, and all you viewers have to keep this a little top secret until we publish. But we're very excited to finally get this working. So just to let you visualize it in 3D, I've got a couple of videos. So this is the same thing. So we've got bone in blue and blood vessels of the cochlea in yellow, looking at this area of the cochlea from top-down view. And if I play the video, this panel will rotate. And what you can see is these wiggly blood vessels, the yellow lines will come around and travel around in 3D this way and that way. So you can start to see the complexity in 3D. This is another one here, which is going to swing side to side. So again, the very wiggly blood vessels can be seen, and I hope you can see the cochlea turn, like spiral going this way, and blood vessels traveling through the center and now heading towards the outside. So these ones are called radial arteries of the cochlea. Now, one thing that you can really appreciate looking at this image is that we seem to have a lot of different 180-degree turns. And this is a very interesting observation that has been suspected before in 2D, and now we show this in 3D. If we are to travel from one place to another, obvious route is a straight one, and if you laid out a curved path, you would be facing some complaint. So how is it that the cochlea seem to opt in for this wavy, winding curves instead of laying down a straight road? Now, this is something that I don't have an answer for, and I'm not sure if I will in any time in the near future. But one of the suspicion is that when the blood travels through the vasculature, and the heart pumps the blood, we have a pulse, and pulsation is a vibration. And if you recall Peter's lecture, our cochlea is a hypersensitive mechanical device that detects sound vibration. So maybe it is that the cochlea does not want to be influenced by the vibration coming from pulsation because it's really tuned to sensing the vibration from sound. And just maybe the curving the road traffic for the blood vessels might dampen that pulsation flow to support the function of the cochlea. So that's a speculation, and we don't know the truth yet. So now I'll move on to the part 2 of this talk, which is to look at a little bit more in detail of some of these vessels using a microscopy approach. So here is a human cochlea, and mind you, this is a pea-sized organ, and this is a panel or photograph panel taken from this area here. And it's very opaque because it's made of tissue in the bottom and difficult to visualize. So we use a special technique called tissue clearing, where we use a series of different chemicals to change the cochlea tissue and alter their property to make them more transparent. So cochlea is still here, but you can see through the grid. And this helps us visualize cochlea with microscopy technique in quite a large volume, just as an example here. So using this technique, as well as conventional confocal and different microscopy technique, we have investigating a selected part of the cochlea. And first I'm going to show this winding blood vessels visualized using low magnification. And this is example from AMRF magnification image. And what we can do with microscopy that we can't do with micro-CT is that with microscopy, because microscopy is a light-based technique with different colors, we can use different stains to highlight different features associated with the blood vessels. So in this panel, we have green color, which stains or colors in arteries, in arterial walls. And we have a blue, which is stains nucleus of small cells that constitute our body. And we have a red stain, which kind of stains the borderline for the bone. So using these stains, what we can see is a bony canal or the tunnel in which the blood vessels travel, and blood vessels themselves in green. And remember that there are a lot of windy curves that we saw in microcity. So if we imagine that traveling through a bony tunnel, and if you take a cross section here, then you will have a tunnel and cross section of individual in these rows, as you see in this microscope image. And I think that's what we are seeing here. This is another example, again, in microscopy and staining showing the cavity inside the bone. So this is the tunnel and the vessels. So you can clearly see a big tunnel and much smaller vessel forming a winding curves within this space. And at the higher magnification, we can even see a little bit more. So we can see smooth muscle cells wrapping around the vessel, and this is a feature of an arterial vessel, so we can confirm these are arteries by nature. We can see some endothelial cells, which are the cells that make up the very inner lining of the blood vessels, and some of the blood cells that travel inside. And with this technique, we are using different other dyes as well to study some other features of the blood vessels, as well as trying to quantify or measure the exact size of the vessels within the bony cavity to understand more about the blood vessels. Now, one of the really quite critical destinations for blood vessels is to reach spiral ganglion neurons. Now, as Peter mentioned in his talk, spiral ganglion neurons are these very specialized auditory neurons that play a really important role in receiving the hearing sound vibration information and sending it right down to the brain. And it's not only important for people with hearing capabilities, but also people with cochlear implant. The functionality of cochlear implant depends on spiral ganglion neurons being able to receive signals from the cochlear implant. So for us to understand how spiral ganglion neurons can maintain its health and how we can support it is really quite critical. And unfortunately, spiral ganglion live in this deepest part of the cochlea where they have quite a nice housing complex. So it has been difficult to study. So that's the area that we are focusing on in particular with this project. And this is a picture showing this area. So yellow dot shows a special bone cavity in the bone. And we have 30 or so spiral ganglion neurons in this particular image living in this yellow housing complex. And green color shows the blood vessels that's in the housing complex with the spiral ganglion neuron. So again, with a higher magnification, we've got neurons, and very close to neurons and right next to them, we have a very, very small capillaries of the inner ear in these areas, which we are trying to reconstruct again in 3D in a little bit smaller area only, but trying to understand their connectivity in 3D and quantify or measure the density and size of these vessels in this area to understand how much blood flow, for example, that the spiral ganglion may receive. In an ongoing work, we are also investigating a protective feature called blood-Rabinowitz barrier that exists in this area based on previous studies and any signs of that in these vessels. So just bringing it all together, we are combining different approaches to make a very comprehensive map of blood vessels in the cochlea. And in ongoing work, we are actually trying to compare how these features of the blood vessels change or stay the same at different age. So we can understand how blood vessels might change from young to older throughout the life course. So just to finish up with, this is a very fundamental research to make the map and we all know map has so much use. But where does this leave us in how do we intend to use them? Because that's the important part. So just to finish off with future direction, this AMRF funded project is going well, and the data coming out is a fundamental map. And this opens up a number of opportunities, and I'd like to just share a few with you. So based on that map, can we model the blood flow inside the cochlea? And what does it mean for the flow that we have these branching and narrowing, as well as say these winding rows? And because these vessels are not only important for blood cells and immune cells, but also therapeutic molecules such as drugs that we administer through oral tablet or intravenous injection, they also travel around our body through blood vessels. What's the implication? Can we model that? How things might get into the cochlea, and how does it get removed from the cochlea through the blood vessels? And that is something that we still have no idea in the human cochlea. Secondly, Peter's mentioned about the tonotopic map of the cochlea and how different areas of that spiral turn are responsible for high frequency pitch or low frequency pitch. And we can now combine the map with our knowledge about how different part of the cochlea is responsible for different pitch. So can we correlate if, for example, we have a blockade of this vessel here, which affect the function of this area of the cochlea, can we then predict how that might manifest in terms of loss or impairment of our hearing in a certain frequency? Because our hearing sensitivity at certain frequency range is something we can currently measure using a diagnostic tools like audiometry. By having our knowledge about the vasculature, can we understand more from our current diagnostic tools? And finally, can we perhaps come up with a novel tools or a medical device that target looking at blood vessels for understanding what really is the cause of hearing loss, in particular for some of the hearing loss like sudden onset hearing loss. And in this regard, Peter will talk briefly about, but we are thinking of a device that may, for example, go through the ear canal and target the cochlea through the ear canal pathway. And with this fundamental work ongoing, can we target measuring blood flow inside the cochlea with this kind of future device? And that would be a whole new way that we might be able to understand the pathology underlying human sensory hearing loss. So I will stop there. Thank you so much for listening, for everyone for joining us tonight. I'd like to just acknowledge our the project team again, as well as Suha and Poi, who have contributed to some of the imaging. And I'd like to thank Organ Medical Research Foundation for funding this research and giving us the opportunity to work together to address this very important research gap. Thank you very much.
So welcome back. You would have heard all the exciting research that Dr. Suzuki Kerr is doing on the vasculature. And she would have also talked to you a lot about the fact that it's a very, very difficult organ to work with. And you have to have some particularly special techniques to be able to study the inner ear. And I want to go on now and bring some of the work that we're doing together and sort of talk a little bit more about the program of work we're doing to look at developing therapies and delivering therapies to the inner ear and translating that into human treatments. So the first thing I want to talk a little bit about is the barriers that there are to inner ear therapeutics. And Dr. Suzuki Kerr would have talked a bit about this in her presentation, in which she talked about the fact that it's embedded, well, the inner ear is embedded in the temporal bone deep inside the skull. So it's actually quite difficult to access for any medical intervention. certainly for surgeries, but particularly also for the delivery of therapies and for, say, the biopsy or being able to actually know what's going on inside the inner ear that's causing the disease in an individual. So one of the things that we've been doing and been working on for quite some time is focusing on a part of the inner ear called the round window. And this is a little membrane that separates the middle and the inner ears. It separates the inner ear, in particular the basal part of the inner ear. And it can be accessed through the eardrum. So if you just look at this, if you're looking at this particular slide, if you look on the left-hand side, you'll see what is viewed down the ear canal. So if you had a microscope or an otoscope, you could look down the ear canal and you'd see the eardrum, which is that disc that you see, that translucent disc. And behind that disc, you'll see what is labelled here as number 3, and are the small bones. Those little bones that I talked about at the beginning, the little bones that transfer the sound across the middle ear into the inner ear. So they're attached to the eardrum. You see a number at the point there. It's a bright point number 4, and so the movement of the eardrum would be moving those bones backwards and forwards. Now, if you're a skilled surgeon, you'd be able to take a little tiny knife and you'd be able to cut around the eardrum and pull it out. And behind that, you'd look into the middle ear and you'd see those bones in real life. And they're shown here as M and I and S, and those are the little bones that connect and go into the inner ear, which is going right into the screen, if you like, right into the bone. And what you see on the bottom part, which is labelled the cochlea, is just the bony wall, the outer wall of the portion of the basal part of the cochlea. And just onto the left-hand side is the round window. It's a tiny little wee window, about 2 millimeters in diameter, that is just a membrane, like the eardrum, not too different in structure to the eardrum. And it separates the inner ear fluids, which is behind the screen there, and the middle ear, which you're looking at in that picture. And if you were, as a surgeon is able to, you can make a small slit in the eardrum, as is shown in the yellow ellipse there. And if you went through at that point, you would end up directly onto the round window. So you can go down the canal and access the round window. And so a lot of the work which we're doing currently is to think about how to deliver drugs, and deliver any therapies into the inner ear through that round window membrane. So we're working on a device. It's a handheld device that would be able to inject and also push the drugs through that membrane and into the fluids of the inner ear. We're only in the preliminary stages, but the idea here is that it would be quite a simple procedure. It could be done in the office setting with a skilled surgeon, obviously, but being able to do what is done clinically in a routine way, for example, inserting grommets or for treating glue ear. And this probe is only 1 or 2 millimeters in diameter, so it's not going to have a huge impact on the eardrum when it's passed through, placed up against the round window, and push the compounds through that. And we talked a little bit about how to maybe understand what's going on inside the human inner ear. So we're thinking maybe you can use that round window, that membrane, as an observatory, a window into the ear. So you can look through it in some way, maybe not visually, but using imaging devices and other sensing devices, which we're looking at developing. And you'd be able to look through that membrane using a similar probe to what I talked about on the previous slide. And you could then look at the structures behind it and also look at the composition of the fluids inside the inner ear to be able to get some estimate or understanding of the disease process that might be going on and how your drug is being delivered and what sort of concentration it might be and so forth. So this is very, very exciting work, which is being led predominantly by Dr. Suzuki Kerr and with a group of physicists and one of our students here, Vanshika, who did some of the preliminary work and won a prize. This work is obviously involves a lot of people with backgrounds as physicists, biologists. We have a number of clinicians who are helping us to make sure that this translates into human studies, and also we have a Māori focus in particular, and we're being very supported by a team of Māori specialists in this area. And graduate students as well, a lot of opportunity for graduate students in this work. I've just shown here two, Poyi Lu and Suna Han, who are directly involved in some of this work through their PhDs. And I'd like to really acknowledge the tremendous support we get from a lot of our funders, and in particular I'd like to acknowledge the wonderful support from the Auckland Medical Research Foundation, who have supported our research into hearing and ear disease over many, many years. So thank you.